In 1972, it was claimed that over 90% of our DNA was worthless junk. But copying and maintaining this much DNA is energy expensive. So what could this DNA possibly be for? It's true that only 1 to 2% of our DNA actually codes for proteins. Transcription factors are proteins that bind to sections right in front of our genes called promoters to turn these genes on and off. Genes that are turned on are then transcribed into pre-mRNA. Introns are non-coding sections of pre-mRNA that are cut off, allowing for the remaining exons to be mixed and matched to form different proteins from the same gene. Other non-coding RNAs form the ribosomes and the transfer RNAs. Other non-coding sections of DNA make up structural elements, like the telomeres, which prevent loss of genetic material during cell division and aging, and the centromeres, which help segregate chromosomes during cell division. But where does all this extra DNA even come from? Junk DNA comes from many sources, including transposition, genomic duplication, and viral insertions. Changes to the DNA sequences of these regions over time can cause them to lose their ability to produce proteins. Some junk DNA can be altered to regulate the expression of genes, potentially preventing an innumerable amount of genetic disorders. This paves the way for a new era of personalized treatments and diagnostic tools. I hope you've learned that junk DNA is a misnomer used for non-coding DNA. These important sequences regulate gene expression, form parts of the ribosomes and tRNAs, include structural elements, and may someday be used to cure disorders. I don't know about you, but this junk seems pretty useful to me.